The overall goal of this procedure is to provide an overview of the transcriptomic changes in bacteria incurred under a specific condition. This is accomplished by first growing and harvesting bacterial cells under a specific condition. Next, total RNA is isolated from the harvested cells, and cDNA is made from the RNA. Then, the cDNAs are labeled and hybridized to in-house made microassay slides. Finally, the slide is washed, after which the slides are scanned and analyzed. Ultimately, the microarray is used to show the transcriptomic changes in bacterial cells under the specific condition.
The main advantage of the DNA microarray technology over other existing methods for studying gene expression like qPCR or northern blotting is that it provides a global overview of all the transcriptomic changes incurred under a specific condition. It is fast, reliable and relatively cheap. Demonstrating the procedure will be Mohammed Afzal and Irfan Manzoor, PhD students from my laboratory in molecular genetics.
After thawing S. pneumoniae D39 cell pellets and resuspending in 400 microliters of TE according to the text protocol, prepare the following in RNA-free screw cap tubes: 0.5 grams of glass beads, 50 microliters of 10% SDS, 500 microliters of previously prepared phenol chloroform, and the resuspended cell pellets. To break the cells, place the tubes into a bead beater and homogenize twice for 60 seconds each.